Hi. In this video, we continue with the respiratory physiology. We have finished with the ventilation, perfusion, and other parameters regarding the respiratory physiology. Today, we discuss about the transport of gases, oxygen and carbon dioxide. So, first, we start with the transport of the oxygen. So, as you know, the most important compound that is important for the transport of the oxygen is the hemoglobin, the Hb. So, at the pulmonary end, most of the oxygen combines reversibly with the hemoglobin and a small amount dissolves in plasma. At the tissue capillaries, oxygen is released from the hemoglobin. Okay, so how much is like combined with the hemoglobin? Around like 97%. In the physically dissolved form, around 3%. But very importantly, this 3% is very important for the partial pressure of oxygen. Okay, both anywhere in arterial or venous blood, everywhere only this 3% the dissolved form contributes for the partial pressure of oxygen. The bound form or the reacted forms cannot contribute towards the partial pressure of oxygen. So importantly, if there is like a decrease in hemoglobin, like in the case of anemia, or decrease in the RBC number, what will not happen? The partial pressure of the oxygen will be normal, no decrease. Okay, so partial pressure of oxygen will be decreased. In the case of hypoxemia, where you go to high altitude or something, you can have a decrease or less oxygen coming to the blood due to emphysema, etc. You can have a decrease in partial pressure of oxygen. But a condition in which the hemoglobin or RBC is decreased, like in the case of anemia, what will not happen? No change in PO2. Okay, important for you to remember. Okay, so let's see what are the factors that controls the oxygen transport and how is the hemoglobin oxygen dissociation curve behaves. Okay, so first point before going to the oxygen hemoglobin dissociation curve, let's discuss about the oxygen content of the blood. Okay, so the oxygen content of the blood means it's not the saturation or the percentage, it is the percentage of the oxygen in 100 ml. It's a value like we normally say it as like 19.4 ml of oxygen is present in 100 ml of blood. 100 ml of blood. Okay, 100 ml of blood. Which blood? 100 ml of arterial blood. Arterial blood. Okay, whereas the venous blood contains only 14.4 ml of 100 ml of venous blood. Okay, venous blood. So this is the meaning like oxygen content. How do we derive at this like a 14.4 and 19.4? It's because then we consider the normal hemoglobin concentration as 15 grams per deciliter. 15 grams per deciliter as a normal value. Normally considered as 14.4 gram per deciliter as the ideal value. Okay, but it is slightly more in males and slightly less in females. You know already those points. Okay, so each gram can carry 1.34 ml of oxygen. Okay, 1 gram of hemoglobin can carry 1.34 ml of oxygen. So if you multiply by that with the 15, you get like 20.1 ml. Okay, so this 20.1 is present if all this hemoglobin is carrying 100% saturation. But in our body you don't get like 100% saturation. You get like around like 97-95% saturation, 95 to 97% saturation. You don't get like 100% saturation. That's why this, instead of 20.1, how much you get? Like 19.4 ml per deciliter. Okay, that's the amount of oxygen that can be carried in the arterial blood. Okay, and on an average, the blood that is delivered to the tissue is around like 5 ml per deciliter. 5 ml per deciliter. So that means the venous blood will have about 14.4 ml per deciliter. Okay, per deciliter. Okay, there is the oxygen content in the venous blood. That's what is being shown. So the amount of oxygen delivered is around 5 ml. Okay, but during exercise, what happens? The muscle cells use oxygen at a rapid rate, which can cause the muscle interstitial fluid, the partial pressure of oxygen, to fall from normal 40 to 15. So very low. At this low pressure, the oxygen carrying capacity, oxygen content of the venous blood will be about 4.4. This will change from 14.4 to 4.4. So instead of giving 5 ml, Okay, 19.4 minus 4.4 will give you 15 ml. Okay, so 15 ml per 100 ml of blood, per deciliter of blood is delivered during exercise, during the exercise. Okay, so that is, it is increasing the capacity of oxygen delivery, so by changing the property of the hemoglobin. Okay, so this 15 ml is the quantity of oxygen actually delivered to the tissues by 100 ml of blood. Okay, not only that, not just 100 ml, the increase in blood flow will also happen. Okay, that means the cardiac output will be increased during exercise. So oxygen delivery to the tissue equal to cardiac output multiplied by the oxygen content of the blood. Okay, so oxygen content of arterial blood decreases as hemoglobin falls, but oxygen saturation and arterial PO2 do not, as PO2 is determined by the dissolved oxygen. That's the first point that we discussed before. Okay, so very important point for your exam point of view. Remember that partial pressure of oxygen or the oxygen saturation does not decrease or change when there is a fall in hemoglobin and anemia. Okay, so now let us see how the property of the hemoglobin changes in different situation. Okay, so let's look at the oxygen hemoglobin dissociation curve over here. Okay, so the important points that we have discussed now over here is like 19.4. Okay, 19.4 ml. Okay, volume percentage. Okay, the volume percentage means ml per deciliter. Okay, another point that we have seen over here is the 14.4 ml per deciliter. Another point that we have seen over here is the 4.4 ml per deciliter. Okay, so this is your arterial content, arterial, venous in the normal, and this is the venous in exercise. Okay, in exercise. Okay, and at the same times, okay, so let's look in the arterial blood. In the arterial blood, what is the approximate partial pressure of oxygen? Approximate partial pressure of oxygen is around like 95 or 90s. Okay, so that is around this value is the partial pressure of oxygen in the arterial blood. In the venous blood, it is around 40. In the venous blood during the exercise, it's around 15. Is around 15. Okay, so these are the various values you can see over here. Okay, so let's put it as 95 over here. Okay, so at this point you get like, at this point you get 19.4 ml. Okay, and how much is the percentage saturation? Approximately 97 percentage saturation. Approximately 97 percentage saturation is seen. Okay, but by the time the partial pressure of oxygen falls to around like 40, what happens to the partial pressure, what happens to the percentage saturation? It becomes around 75 percentage. Okay, it becomes around 75 percentage, and the oxygen content of the blood will be around 14.4 ml. Okay, so this is what you see in this 40. Okay, at 40 you will see around 75, and then around 14.4. Okay, by the time it falls down to around like 15, what happens? You will get like around 25 percentage saturation. You will get around 25 percentage saturation. By the time it falls to 15, you will get around 25 percentage saturation and 14.4 ml of air in the venous blood during the exercise. During the exercise, that's what you see. Okay, so this is like the lowest partial pressure that can be attained. The 15 is the lowest partial pressure that can be attained by the tissues during exercise. During exercise, it can fall as low as 15. Okay, so this is the hemoglobin, oxygen hemoglobin dissociation curve. So you can see the first part from this point to here. Okay, so that is like 97% saturation to 75. This is considered as the flat portion. You don't see much of the dissociation. It's hard for the oxygen to dissociate because only around 5 ml is dissociated. Only about 5 ml of is dissociated, or like around 25% is dissociated. But the second portion you can see over here, which is the steep portion over here, from 75% saturation to 25% saturation, or from partial pressure of 40 falling down to 15. Okay, you can see a huge fall in the uh, dissociation, huge fall in the dissociation, or very much dissociated. So how much approximately dissociated? Around 50%. From 75% to 25%, how much is dissociated? 50% is dissociated. If you look here, how much is dissociated? Around 10 ml is dissociated. That means during exercise, a total of 15 ml per deciliter is dissociated. 15 ml of oxygen per deciliter is dissociated. That means oxygen can be delivered to the tissues. Okay, so why the hemoglobin have this property? In the beginning, even though there is a huge change in the partial pressure of oxygen falling from around 95 to 40, a difference of approximately 60, you are only dissociating only about 25 percentage. But when the hemoglobin changes its property, and when it falls, the partial pressure of oxygen falls below 40, its dissociation is very much increased. Okay, so this property change in the hemoglobin is associated with the structure which you have studied in your biochemistry. The hemoglobin have like difference in the amino acids that can interact with each other and changes its property from taut to relaxed state, R state to T stage, and this relaxed and the taut forms of the hemoglobin can easily cause the dissociation of the, that's why you get a steep portion. But after this 15, again you get a flat portion. It's not easy to, below 15 it's not easy for the hemoglobin to dissociate the oxygen. Okay, so if you just take like hemoglobin as 4 molecule. Okay, so this is the first oxygen. Okay, and this is the second oxygen, third oxygen, and the fourth oxygen. So when it is four, all the four are there, it's difficult to release this oxygen, the first oxygen to release. But once the first oxygen is released, okay, then what happens? It becomes very easy for these two to get released. The second and the third one to get released. This will be released. Okay, but the last one, it becomes very hard for the hemoglobin to release the oxygen. The last oxygen is very difficult to release, same like the first oxygen. The first oxygen is also, the first 25% is difficult to release, and the same like the last 25% is very difficult to dissociate. Okay, so when it is the last 25% to dissociate, when the partial pressure of oxygen falls below 15, when the partial pressure of oxygen falls below 15, what happens? In the muscles you have another compound. What is that compound? The myoglobin. So if you draw the myoglobin curve, it goes up to here without any fall, but after the 15, what happens? It falls drastically down. Okay, so this is how, this is not like a sigmoid curve for the blue line that we have shown. This is for the myoglobin. The blue line is for the myoglobin. So this is like a single unit. There is no four units. Okay, this carries the oxygen. Single myoglobin carries one oxygen. So there is no interaction between the other. So there is no release of the other. So this is the last oxygen that is getting released from this myoglobin. Just one of. So this myoglobin molecule, single subunit. So that's why it releases the oxygen after the partial pressure of oxygen falls below this 15. Okay, when the exercising muscles. Until then, is the hemoglobin that can release. So myoglobin will show not the sigmoid curve, instead the parabolic. Okay, instead the parabolic curve, and then after the 15 it falls below. Okay, so let's see the other scenarios in which the oxygen hemoglobin curve can change. Okay, so as we discussed in the anemia, the saturation, this curve over here, this side over here, the hemoglobin percent, the saturation percentage does not change. This remains the same, but that is not the case with the volume percentage. The volume percentages will fall, because for example, this 19.4 we got whenever there is a 15 grams of hemoglobin. 15 grams of hemoglobin. But if in a patient, if it falls down to 10 grams of hemoglobin, what will happen? We discussed like each gram can carry 1.34. So multiplied by 10, how much is the maximum it can carry? It can carry only 13.4 ml of oxygen per deciliter. This is the maximum of oxygen that can be carried in a condition where the patient is having only 10 gram of hemoglobin. Okay, so that is important to understand. When the hemoglobin decreases, the volume percent, the amount of oxygen that can be carried, the whole oxygen content of the blood falls. Okay, so how the curve will look like? Okay, if you take only the oxygen content over here, okay, on the other axis, like the hemoglobin saturation axis, no change will be seen. So that's why you can see over here, there is the oxygen plus hemoglobin. This is with the 7 grams per deciliter. Okay, so this is with the anemia. In the anemia, so the curve will fall down. Okay, it will shift to the right and it will fall down. Okay, so this was showing like around like 19.4, but in this patient, because it is only 7, it is like below 10. Okay, below 10, say maybe like 9. Okay, approximately, only that's the maximum hemoglobin saturation, like maximum oxygen carrying capacity that can be attained, not like 19. Okay, so it will be very low and it will be shifted to the right. And what happens, this the dotted line, the oxygen plus hemoglobin with 50% carboxyhemoglobin. Carboxyhemoglobin means what? Carbon monoxide. Okay, CO, not CO2. Carbon monoxide is attached to hemoglobin. When the carbon monoxide is attached to the hemoglobin, what happens? It becomes very difficult for the oxygen to get released. You cannot release the oxygen from the hemoglobin. The affinity for the oxygen to the hemoglobin is increased. Okay, the affinity for the, so what will happen, even though the partial pressure is falling, no change in the graph, it keeps on staying at the same. But when the partial pressure falls below 15, then same like the myoglobin, it will fall down. Okay, so until then it cannot release it, that oxygen cannot be released from the hemoglobin until the partial pressure is very low for the oxygen. Okay, so that's why it becomes very difficult for the oxygen to be released in the case of a carbon monoxide poisoning. Okay, so effects of PO2. So at PO2 greater than 90, hemoglobin is almost 100% saturated, almost like 97% saturated, and at 40, about 75% saturated. So this implies 25% of the oxygen bound to the hemoglobin is released during the resting stage. Okay, so condition like vigorous exercise can decrease the PO2 to as low as 15. At this stage, the hemoglobin released nearly 75% of the bound oxygen. Okay, because this the saturation is falling down to 25%, that means remaining 70%, 75% is dissociated. Okay, so that means increased exercise means what? Increased tissue need. In increased tissue need, there will be fall in the PO2, and whenever the fall in PO2, what will happen? There will be increased release of the oxygen. Okay, so PO2 is a major factor that controls the hemoglobin's release of the oxygen. Apart from the PO2, we have certain other things that controls the uh, hemoglobin dissociation curve. Okay, so that is called as the Bohr effect. Okay, so the influence of all other substances like carbon dioxide, which changes the pH, which changes the hydrogen ion concentration, and which another substance that can be changed by that is the 2,3 bpg concentration in the RBC and the temperature in the RBC. All this will cause what? A right shift to the curve. Okay, so that means increased release. So whenever there is an increase in the oxygen content, increase in the carbon dioxide content, increase in the pH, decrease in the pH, or increase in the hydrogen ion concentration, or increase in temperature, and increase in 2,3 bpg. Okay, so will cause what? A shift of the curve to the right. Shift of the curve to the right. That means there is, whenever it is shifting to the right, there is more carbon dioxide, so more oxygen being released by the influence of the carbon dioxide. That is what is called as the Bohr effect. Okay, that's what is referred to as the Bohr effect. Okay, so what happens over here? So when the, for example, this is your RBC. If the carbon dioxide enters, okay, what are the things that it do? Okay, so hemoglobin is here with the oxygen. The carbon dioxide will react with it and cause carbaminohemoglobin. Okay, hemoglobin with carbon dioxide is referred to as what? Carbamino. Carbaminohemoglobin. Okay, because it is binding to the globin part, not to the heme part. The carbon dioxide binds to the globin part, not to the heme part. Carbaminohemoglobin. This carbon dioxide binding to the globin part will cause the oxygen to go out. Okay, so this is one effect, direct effect of the carbon dioxide on the hemoglobin to release the oxygen. Okay, so we are discussing the Bohr effects. Okay, the Bohr effect, the influence of the carbon dioxide. Another thing is like when the carbon dioxide comes in, what happens? The carbon dioxide will react with water. Okay, in the presence of carbonic anhydrase enzyme, and causes what? The formation of H2CO3. Okay, H2CO3 will get dissociated into H plus and HCO3 minus. HCO3 minus. Now, what happens with this H plus? What are the things that is done by this H plus? Okay, so the H plus that is coming from the carbon dioxide that is coming in and forming the H plus. H plus will bind to the hemoglobin. HB. Okay, the H plus is binding to the hemoglobin, HHB, which will cause the oxygen to be released. Okay, the hemoglobin was with the oxygen before, and this oxygen will be released out to the tissues. Okay, the HHP. Okay, so this is another influence of the Bohr effect. So where do we get the hydrogen from? From the carbon dioxide. Another influence of this hydrogen ion is what? This hydrogen ion stimulates the mutase enzyme. The mutase enzyme. The mutase enzyme causes the conversion of 1,3-BPG to 2,3 BPG. Okay, 2,3-BPG. And the 2,3-BPG binds to the beta chain. Which chain? The beta chain, not to the alpha chain, to the beta chain. Okay, beta chain of the hemoglobin. And what will happen in the presence? So that means in the presence of carbon, when the carbon dioxide comes in, carbon dioxide forms H plus. H plus increases the 2,3-BPG. It's the byproduct that is formed in the by stimulating the mutase enzyme by the hydrogen ion in the glycolysis pathway. So 2,3-BPG binds to the hemoglobin. Okay, which chain of the hemoglobin? Beta chain of the hemoglobin, and it was with oxygen. Now when the 2,3-BPG comes in, what will happen to the oxygen? The oxygen will go out. The oxygen will not be with the hemoglobin, and the oxygen will go out. Okay, so this property is used by the mother and the fetus. The mother have beta chain, but the fetus have the gamma chain. So what will happen? The gamma chain cannot bind with 2,3-BPG. So the mother's oxygen will move from the fetal mother's hemoglobin, the HbA, to the fetal hemoglobin. So we call it as the mother's hemoglobin is called as the HbA, or the adult hemoglobin, and then it goes into the fetal hemoglobin. The oxygen moves from here to here because this binds with 2,3-BPG. Okay, 2,3-BPG is phosphoglycerate. Okay, so whereas the HbF, which contains the gamma chain, here it contains the beta chain. So 2,3-BPG will kick out the oxygen from here, and the fetal hemoglobin can take up this oxygen. So that's one advantage of having the 2,3-BPG that can easily. And in our body, whenever there is an increased carbon dioxide will increase the hydrogen ion. Increased hydrogen ion will increase the mutase and cause the increased formation of 2,3-BPG, and then it causes the kicking out of the oxygen. So beta will be like bound with a 2,3-BPG, and the oxygen can go out to the tissue. So all these phenomena will cause the right shift. Opposite of this, whenever there is less carbon dioxide, less hydrogen ions, less temperature, or less 3-BPG, what will happen? The curve will shift to the left. That means the oxygen will be held with the hemoglobin. It will not get released to the tissues. Okay, so that's what happens in the case of when there is carbon dioxide being decreased. So all this effect, influence of the carbon dioxide on the oxygen carrying capacity, oxygen delivering capacity, is called as the Bohr effect. Okay, the opposite is called as the Haldane effect. The influence of the carbon dioxide release by oxygen. Okay, how the oxygen influences the movement of the carrying of the carbon dioxide is called as Haldane effect. So now let us discuss about the carbon dioxide transport. Okay, so then we will understand. Okay, so transport of carbon dioxide. Okay, so the major form in which it is being transported, or around like 72%, is as bicarbonate ions in combination with and also in combination with blood proteins, mainly hemoglobin, around 20%, and in plasma, how much percentage is carried in plasma? 8%. So the majority is carried as 72%. Okay, so that means carbon dioxide reacting with water in presence of carbonic anhydrase. Okay, so hemoglobin that released oxygen binds more to readily to carbon dioxide than the hemoglobin that has oxygen bound to it. That is called as the Haldane effect. Haldane effect is the influence of the oxygen release oxygen on the carbon dioxide carrying capacity of the blood. Okay, so in order to understand this important factor, that like how the carbon dioxide is transported, let's discuss this question. Okay, so a new drug was being developed to treat restrictive lung disease. It is targeted a newly discovered intracellular target inside the fibroblast that limits the deposition of collagen, reducing the rate of fibrosis. The drug was abandoned after early clinical trials with mice because it was found to cause a precipitous and fatal drop in pH. Okay, so no one had foreseen that it would markedly inhibit the carbon dioxide carrying capacity of the venous blood. Okay, so that means this drug is causing a major decrease in the carbon dioxide carrying capacity. So which of the following mechanism is most likely affected by this newly by this net drug? Okay, so the question is asking you what is the most important markedly inhibit the carbon dioxide carrying capacity. So that means what is the most important carbon dioxide carrying capacity, as we have discussed, as bicarbonate. The carbon dioxide 72% of the carbon dioxide has to be transported as bicarbonate. So decreased solubility of carbon dioxide across the endothelium. No, that is not the reason. Okay, increased amount of dissolved plasma carbon dioxide. No, dissolved carbon dioxide, that to increased. No. Okay, inhibition of RBC, carbonic anhydrase. Very good. Okay, so inhibition of RBC, carbonic anhydrase. Carbonic anhydrase is required for the transport of carbon dioxide as bicarbonate. So this is your answer choice. So if the drug is blocking this one, what will happen? You cannot carry the carbon dioxide. Inhibition of carbaminohemoglobin. No, how much percentage is it? Is only 20 percentage, not the 72 percentage. Okay, so it's only 20 percentage as carb aminohemoglobin. Okay, so inhibition of H plus HB. Okay, no, not that one, not this one. So this is your answer choice. Okay, when you inhibit the H plus hydrogen binding to the hemoglobin, it changes the oxygen delivery, not the carbon dioxide problem. Okay, hydrogen is binding to the hemoglobin will cause the kicking out of the oxygen. Okay, so the oxygen delivery to the tissue will be reduced if you inhibit the hydrogen binding to the hemoglobin. Okay, so hope you understood the importance of this 72 percentage. Okay, so let's see in detail of those. Okay, so what happens in the tissue level? Okay, so this is what is shown in the tissue level. So in the tissue levels, what happens? The carbon dioxide comes in, and some of the carbon dioxide will bind to the hemoglobin, approximately like 20 percentage. Okay, almost like the remaining percentage is like reacting with water in the presence of carbonic anhydrase to form H2CO3, and the HCO3 minus is shifted out in exchange for chloride ions. So this exchange, bicarbonate to chloride exchange, is called as what? The Hamburger phenomena. Okay, also this is caused by the anion exchanger. anion exchanger Anion exchanger. The most commonly used anion exchanger is referred to as the band protein. Okay, band 3 or band 4.1, etc. Okay, so these band proteins are the one that is responsible for the bicarbonate to chloride exchange. A mutation normally seen in this band protein will result in spherocytosis. So this topic we will discuss more when we discuss about the RBC structure in the hematology chapter. Okay, spherocytosis. Just to understand, the band proteins are responsible for the anion exchange. Okay, so bicarbonate is going and the chloride is coming in. One significance of this chloride coming in is what? Along with the chloride can attract little bit of sodium, and when this it attracts a little bit of sodium in, it attracts also a little bit water along with that. So this will cause a little bit of swelling of the RBC. Okay, the RBC will swell up little bit. So when you see the hematocrit, okay, the hematocrit, so normal say is like 45 percentage. Okay, so slightly there is a slight difference between the hematocrit, that means the percentage of RBC that is sedimented when you centrifuge the blood. Okay, so that the amount of RBC, the percentage of RBC that you see when it is swelled up, it will be slightly more. So in which blood do you think there will be more hematocrit? Definitely, because that when the carbon dioxide is coming in, that is in the venous blood. When the carbon dioxide comes in, what is coming in? The chloride is coming in. When the chloride comes in, it comes along with little bit of sodium and water, which cause little bit swelling of the RBC, and the hematocrit of the venous blood will be slightly more than the arterial blood. Okay, the hematocrit of the venous blood will be slightly more than that of the arterial blood. Maybe one question you can get from this concept, okay, about like the moving in of the chloride. Okay, so now what happens in the oppositely in the when the blood reaches to the lung? When blood reaches to the lung, what happens? The bicarbonate comes in, and what goes out? The chloride goes out. Okay, and the bicarbonate will react with the hydrogen ion again in the presence of carbonic anhydrase, will cause the conversion of the H2CO3 into carbon dioxide and water. The carbon dioxide from here and also from the hemoglobin is released. Okay, this happens when the oxygen comes in. Okay, so from the here what is coming in? The oxygen is coming in. The oxygen causes this shift. Okay, so that is what is referred to as the Haldane effect. When the oxygen is coming in, it causes the movement of the carbon dioxide, the Haldane effect. So oxygen comes in, the carbon dioxide moves out, and carbon dioxide goes to the lung for the exchange out. So again, what is required? The carbonic anhydrase is very important for this phenomena also. Okay, so let's discuss about the Bohr effect and the Haldane effect. Okay, the Bohr effect and the Haldane effect. Just now we have discussed about the Bohr effect and the Haldane effect. Okay, let's see like the magnitude of the Haldane effect. Previously we have seen discussed about the Bohr effect in the case of oxygen hemoglobin dissociation curve. We have seen how the shift can happen. Okay, right shift versus the left shift. Okay, about the Bohr effect that's happening because of the Bohr effect. Okay, so now because of the Haldane effect, how the oxygen, similar to that of the oxygen hemoglobin dissociation curve, we have the carbon dioxide blood dissociation curve. Okay, so the carbon dioxide dissociation curve. Okay, so in the blood we have seen the arterial blood, how much is the oxygen content? We have 19.4. How much is the carbon dioxide content? Okay, so it's about 48. In the venous blood, what happens to the oxygen content? It's around 14.4. The venous blood will take the carbon dioxide and becomes 52. So the amount of oxygen delivered over here is about 5 ml. The amount of carbon dioxide being taken out from the body is around 4 ml. Okay, so the ratio of these two is called as the respiratory quotient. Okay, or the respiratory quotient or the respiratory exchange rate, which is 4 divided by 5 carbon dioxide over the oxygen value. Okay, carbon dioxide delivered by the oxygen value equal to 0.8. So this is your normal exchange rate. Okay, normal oxygen respiratory exchange ratio or the respiratory quotient. Okay, we call it as the respiratory quotient. Okay, which is equal to 0.8. Okay, so this 0.8 is very essential to be used in an equation called as the alveolar gas equation. The alveolar gas equation is used for calculating the alveolar partial pressure of oxygen, the capital A, which is very essential for the clinical purpose to understand the AA gradient, which we have discussed before. Okay, so AA gradient. So this will be given in the ABG value. Arterial blood gas value will give you the arterial oxygen content or the arterial partial pressure of oxygen. But the partial pressure of oxygen in the alveoli, okay, this is in the alveoli, it is difficult to get from the ABG value, so you need to use this for the calculation. Okay, so the inspired oxygen, the partial pressure of inspired oxygen, which is around like 149. Okay, 159 is there in the atmosphere, and then after mixing with the humidifying with the water vapor, what will happen to that? It will come down to around like 149. The inspired oxygen will be around 149, and then the partial pressure of arterial carbon dioxide, which is around 40. Okay, and the respiratory quotient is equal to 0.8 approximately. So if you solve this equation, it will come the alveolar oxygen is around 104. Okay, so that's how you get this calculation. So maybe in the question you may have to calculate the alveolar oxygen and then use it to understand the AA gradient, if the AA gradient is increased or if the AA gradient is normal. Okay, dependingly you can get the differential diagnosis between whether it is a lung or the cardiovascular problem compared to that of the atmospheric oxygen content decrease causing a hypoxemia. What is the reason for an hypoxemia? Okay, okay. So with this like information like this 48 and uh, 52, okay, for the carbon dioxide, let's now see the Haldane effect. Okay, supposing if the partial pressure of oxygen is kept at 100, okay, just like 100. Okay, so this is your arterial blood. Okay, this is your venous blood. So how much it would have been like carried? So this is like 48. Okay, and at 40 if it is like 48. Okay, and at 45 it will be around 50. Okay, that means if the partial pressure of oxygen is not being changed, the only 2 ml can be delivered, 48 to 50. Also, if you just check, if the partial pressure of oxygen is just kept static at 40, how much you can deliver? Say from 50, at 40 it will be around like 50, and at 45 it will be at this point say around 52. Okay, then also over here how much you can deliver? 2 ml only. But because from this point to this point, what you change the partial pressure of oxygen in the arterial blood. The partial pressure of oxygen is around 100, PO2 is O2 is around 100, and PVO2, the venous oxygen, is about 40. Okay, so what will happen? It will go from 48. By the time it reaches to here, it is not 50, instead here, because the partial pressure of venous oxygen is changing, it reaches to around 52. That means from here to here, how much you can deliver? 4 ml can be delivered. Okay, this change is happening because of a change in the partial pressure of oxygen. From this point to this point, the partial pressure of oxygen is changing. So that is the influence of the partial pressure of oxygen being changed. So almost instead of like 2 ml being given out, the carbon dioxide carrying capacity is instead of 2 ml, now how much is it becoming? It is doubled to around 4 ml. So that's why the Haldane effect is very significant. Okay, numerically also the magnitude wise, Haldane effect is very significant. It is causing around like 4 ml of oxygen to be delivered. Sorry, 4 ml of carbon dioxide to be expired out. Okay, so that is the influence of Haldane effect. So hope you understood what is a Haldane effect, what is Bohr effect, etc. Okay, so now we have discussed the basic physiological concept that is required for your transport of the gases. Okay, the transport of gases within the blood, that is oxygen and carbon dioxide transport. Now we go to the next topic, that is the regulation of respiration. Okay, so it happened mainly in the pontomedullary region, all the regulation pontomedullary region, where we have the respiratory center. The respiratory center is located in the pontomedullary region. So it composed of several groups of neurons and located bilaterally in the medulla and pons. That's why we refer to the pontomedullary region. Okay, cerebral cortex, cingulate gyrus, the limbic system, and the hypothalamus helps in the regulation. Okay, so that helps mainly for the voluntary control of respiration. So we have two parts, voluntary and involuntary control of the respiration. So when a person loses the voluntary control of respiration, it is referred to as the Ondine's curse. Okay, the name of the Ondine's curse came from the German mythology. Okay, German mythology have this Ondine curse, and in general it's called as the central hypoventilation syndrome. That means the person has lost the ability to do the voluntary ventilation, so he has to always remember to breathe. Okay, so or when he goes into sleep, he has to put it in the ventilator. Okay, it's a respiratory disorder that results in respiratory arrest during sleep. Okay, in the sleep you cannot breathe. Okay, so that's the problem, because you lost the the involuntary control. So in the central hyperventilation, what is lost? The involuntary is lost, only the voluntary remains. So in the Ondine's curse, what is lost? Lost is involuntary. Involuntary is lost. Involuntary control is lost. Okay, involuntary control is lost. Okay, so you still have the voluntary control that is coming from the cerebral cortex, cingulate gyrus, limbic system, etc. Okay, so let's first study about the involuntary and how we can control it like voluntarily also. So the respiratory center includes the ventral respiratory group of neuron, the ventral respiratory group of neuron, and the dorsal respiratory group of neurons. The ventral respiratory group of neurons includes the expiration and inspiration, and this involves mainly the forced inspiration and expiration, the forced inspiration and expiration, and this include the tidal, the normal, the tidal inspiration and expiration, the tidal. Okay, so in the tidal, you need only inspiration to happen. Inspiration is the active process, whereas the expiration is a passive. So that's why you need to control only in the tidal, you need to control only the inspiration. You don't need to control the expiration and inspiration, because that is happening in the forced, where you have the extra muscles coming into part. Okay, so first let us study about the dorsal respiratory group of neurons. Okay, that is your inspiratory center. Okay. In order to control the dorsal respiratory group of neuron, we have the apneustic center, otherwise also referred to as the pre-Botzinger complex. Botzinger complex. Okay, this is the little a newer name for this location where the automaticity is seen. So apneustic center is automatic. Automatic. It fires automatically. The neurons are automatic in nature, keeps on firing. Okay, so they fire the impulses. So from the apneustic center, the information goes. So from the apneustic center, the information goes to the inspiratory center, and it goes to the diaphragm, Okay, and from the diaphragm it undergoes contraction, it stimulates the diaphragm, and you have the inspiration happening. Okay, now how to control this? At the same time, the apneustic center is always firing, it's an automatic. So above the apneustic center you have the pneumotaxic center, which controls the rate and pattern of the breath. Okay, so this is above the apneustic center. So whenever the apneustic center is getting stimulated, it sends stimulatory impulses, it stimulates the pneumotaxic center. In return, what does the pneumotaxic center do? In return, it stimulates in this direction, but in this direction it sends inhibitions. Okay, it inhibits the apneustic center. So when the apneustic center is inhibited, no impulse goes to the inspiratory center and no impulse goes to the diaphragm is blocked, and then what will happen? You get expiration. When the diaphragm is stopped, the stimulation of the diaphragm is stopped, it goes into relaxation, and when it relaxes you get expiration. Okay, so that is the basic rhythm of respiration that you see. Okay, so that is the basic role. So if you cut over here the pneumotaxic center, what will happen? The apneustic center will keep on firing and you keep on having the inspiration. Let's see how in this scenario the diaphragm can be inhibited. Okay, so before that, let's see what is the role of the ventral respiratory group of neuron. Ventral respiratory group of neuron is for the forced inspiration and expiration. So ventral respiratory group does not take part in the basic rhythm of breathing and is almost inactive during the quiet breathing. Ventral respiratory group of neuron is stimulated by the sensory area of the dorsal respiratory group, which is nothing but the nucleus of tractus solitarius. Okay, so when high levels of pulmonary ventilation like exercises are required, okay, what happens? The motor neurons of the ventral respiratory group of neuron stimulate rhythmically both inspiration and expiratory muscles. Okay, so that's the role of the ventral respiratory group of neurons. Okay, so the previous question, when you cut the apneustic center from the pneumotaxic center, okay, what will happen? Then you need Herring-Brewer reflex to happen. The Herring-Brewer reflex. That means there are stretch receptors located in the muscular portions and the bronchi, bronchioles, even in the heart, everywhere. Okay, when overstretched, transmit signals through the vagus into the dorsal respiratory group of neurons. So this signals inactivates motor neurons in the dorsal respiratory group of neurons, thus switching off the inspiration ramp. Why we call the word ramp? Because inspiration happens like this and then suddenly stops. Inspiration happens like this and suddenly stops. Inspiration and then it suddenly stops. This is how the ramp happens. So if you don't have the pneumotaxic center, you cannot control this. Instead, it will keep on going, and then it stops. So what stopped at this point? That is by this Herring-Brewer reflex. Okay, Herring-Brewer reflex is the one that is causing this stop of the inspiratory ramp when there is no pneumotaxic center. Okay, or the pneumotaxic center is like is controlled by the higher brain centers. Then if you keep on inspiring, the stretch receptors will stop the inspiration at a point. Okay, so that is what is called as the Herring-Brewer reflex. Okay, and this also causes increase in rate of respiration. So if you keep on stimulating the Herring-Brewer reflex, what will happen? The rate, you stop the inspiration frequently. Okay, so this reflex is more important in preventing excess lung inflation rather than controlling normal ventilation. Okay, so that's the importance of Herring-Brewer reflex. Okay, so we have seen you need the impulses going to the nucleus of tractus solitarius. That's the sensory part. So let's see how do we get this information to the nucleus of tractus solitarius. That requires the chemical control. Okay, you get the information from the chemoreceptors located in two parts. One is the central and another one is the peripheral. So central chemoreceptors is stimulated by the directly by the hydrogen ions and indirectly by the carbon dioxide, because the carbon dioxide goes. So let's see here in the central. So the carbon dioxide has to come because there is a blood-brain barrier over here. So the hydrogen ions cannot go directly from the blood to the chemosensitive area. Instead, what can go? The if you keep the blood-brain barrier over here, what will come here? Carbon dioxide will come over here. The carbon dioxide will react with water in the presence of carbonic anhydrase enzyme and forms the H plus. The H plus then stimulate the central chemosensitive area, central chemoreceptor areas. Okay, whereas the peripheral chemoreceptors, that is located in the aortic body and the carotid bodies, okay, which is present on the aortic arch, outside of the aortic arch, outside of the aortic arch. Okay, aortic arch, not inside. Inside you have the baroreceptors, outside you have the chemoreceptors. Chemoreceptors requires the chemical content from the capillaries. Okay, so the small capillaries that is outside is supplying that. And also at the carotid bodies, again in the carotid body you have the bifurcation, you have the carotid sinus. Okay, the bifurcation you have. So here outside of these you will see the carotid bodies, not inside. Inside have the baroreceptors. Okay, outside you have the carotid bodies. Okay, and the bodies. And these are the location where you have the peripheral chemoreceptors. Okay, which is stimulated by mainly by the oxygen concentration, a drop in oxygen concentration, and also stimulated to less extent by the hydrogen ions and then the carbon dioxide also concentration, also by lactic acid and other acids like keto acids, etc. Okay, lactic acid, keto acids, etc. can also stimulate the peripheral chemoreceptors, but the central chemoreceptors only by the hydrogen ion coming from the carbon dioxide, not the peripheral hydrogen ions. Okay, so the location of the peripheral chemoreceptors, with this we have studied along with the CVS more in detail. Okay, so fall in oxygen, increase in carbon dioxide, increase in hydrogen ion will stimulate your aortic or the carotid bodies, and from the aortic bodies the 10th cranial nerve, which is the vagus nerve, carries the information to the dorsal respiratory group of neuron, which includes the nucleus of tractus solitarius. Okay, solitarius. So all the sensory information goes to where? The nucleus of tractus solitarius. Okay, then from the carotid body, it is not the 10th cranial nerve, it is the ninth one. Okay, the glossopharyngeal, the glossopharyngeal nerve. Okay, and the branch of that glossopharyngeal that brings the information is called as the Hering's nerve. Okay, take it to the nucleus of tractus solitarius. Okay, so this is how the information goes there, and then the regulation happens. Okay, so let's see how much of this like fall in oxygen is required, how much of the fall in carbon dioxide is required in order to control this. Okay, so this graph, okay, is a composite effects of PCO2, pH and PO2 on alveolar ventilation. Okay, so there are four values given over here. Okay, so you have the PO2, okay, alveolar PO2, and then PCO2, okay, and then you have the pH, and then the alveolar ventilation. So the normal value of alveolar ventilation is 4.2, okay, 4.2 liter per minute. Okay, the normal pH is 7.4, PCO2 is normally is 40, normal value is 40, and PO2 is around like 100. Okay, PO2 is around 100 normal value. Okay, so if you take these values, normal values in the arterial blood, the PO2, the arterial blood, okay, for the alveolar ventilation to happen, how much is the alveolar? So what is the regulating factors? These are the various regulating factors for that. Okay, so what happens to the oxygen change? Okay, so let me put all these normal values. Okay, so here the alveolar PCO2 is like 40 over here, and pH is like 7.4. That means I have to look into this this red line over here, this red line, that is the 100. Okay, and PCO2 is already, I took the PCO2, the 40, 100, and the pH is 7.4, that is shown by the red color. And then alveolar ventilation, how much? So 40, and then it is coming to the 100, and it's around 4.2. Okay, so this is around 4.2. So this is the your normal alveolar ventilation. Okay, so this is the normal alveolar ventilation that you get. So when I change these values, let's see what happens. Okay, so let's see the most important, if you keep these like normals, okay, if you keep these normals, and if you suddenly change this value, if you change this value from 40 to 35, you will see what happens. Says this is your 35 over here. What happens to the alveolar ventilation? It became 0 over here. It became 0. Okay, so the alveolar ventilation will change in this scenario, it will change to 0. That means because the PO2 is normal and pH is normal, there is nothing to stimulate your peripheral chemoreceptors, and for to stimulating the central chemoreceptor you need the carbon dioxide. A minimum carbon dioxide is required, and if the carbon dioxide falls below that minimum value, say from 40 to 35 it has fallen down, what will happen? The ventilation will stop. the alveolar ventilation will be stopped in that scenario. Okay, and not only that, you keep this at like 35 and you drop it down the PO2 say around like 60. Even then what will not happen, even then the alveolar ventilation, even at 60 in this line also, how much is the alveolar ventilation? It's 0. Okay, so even below, like even falling it down to around 60, what is not happening? The ventilation is not happening. That means the peripheral chemoreceptors will be stimulated when the PO2 is falling below 60. Okay, when it falls below the 60 value, then the peripheral chemoreceptors will start to stimulate. So let's see, keep it at like carbon dioxide is very low, and if I put it like 50 or like 40, etc. So what will happen at 35? You can see at 35 you will see a minimum of ventilation of say around like 12, or like if the partial pressure of oxygen is like 40, you can see a minimum of like around like 15. Okay, so the ventilation is very high, okay, in that scenarios. That means because the partial pressure of oxygen has fallen down. So the question will be asking you, in this scenario what has stimulated? It has been stimulated by the peripheral chemoreceptors. Okay, peripheral chemoreceptors have stimulated. Okay, but for example, if you keep the oxygen normal, oxygen is kept at normal. Okay, and pH is also kept at normal at like 7.4, the 7.4, the red line. But if you slightly increase the carbon dioxide, say from 40 to 50, okay, it's slightly increased. Okay, what will happen? It will go very high, say around like 30. Okay, so the ventilation will be around 30, which is like compared to like 4, it's like much higher. Okay, so this high is caused by what? An increase in carbon dioxide. Increase in carbon dioxide will stimulate what? The central chemoreceptors. Okay, is caused by the central chemoreceptors mainly, because the carbon dioxide is causing that. Even though the oxygen content and the pH value are normal, it will get stimulated. Okay, so but if the pH is falling, the pH is falling, what happens? PH can actually stimulate your peripheral chemoreceptor. So you need to drop the carbon dioxide level so low in order to cause a zero ventilation, like around like 25. Okay, so only when it is low, then only you can get a zero ventilation, because at the peripheral chemoreceptors are stimulated when the pH is low, when the pH is falling down from 7.4 to 7.3. So either the pH can stimulate your peripheral chemoreceptors, or the drop in oxygen below partial pressure of 60 can stimulate your peripheral chemoreceptors, or the carbon dioxide can also stimulate, but carbon dioxide mainly go to the central chemoreceptors and get converted into hydrogen, because carbon dioxide reacts with water in the presence of carbonic anhydrase, which is reaching the central chemoreceptors. Okay, so these are the various points that you can remember from this graph. As such, the graph itself does not come in the exam, but it's important to understand these parameters: what stimulates what, and when does the peripheral chemoreceptors get stimulated, when does the central chemoreceptors get stimulated, etc. Okay, good. Okay, so moving on, regulation. From the regulation, let's look into sub abnormal patterns. Okay, first, very important term that you need to understand is the breathlessness or dyspnea or the dyspnea. Okay, dyspnea or the dyspnea. So breathlessness is a phenomena, like it's a feeling like a condition. Okay, because even though there is no problem with the cardiovascular system, there is no problem with the respiratory system, still you can get breathlessness feeling, because you can get it from limbic drive, from the emotions can cause the breathlessness feeling. The cortical drive can cause the breathlessness feeling. Okay, just by increasing the mechanical load on your like just someone like giving a tight hug, or like holding it tight, or a tight dress can give you the breathlessness feeling. Okay, so dyspnea is like actually a feeling. Okay, that you can get this feeling also from the other problems within your cardiovascular or within the respiratory system, like for example interstitial disease, inflammation, infection, fibrosis, etc. Okay, or deflation, pneumothorax, collapse, effusion, etc. can also give that, or like conditions like pulmonary embolus can give that. Okay, all this will cause the vagal lung afferents to send the impulses to the brain and cause the feeling of breathlessness. Okay, so pulmonary fibrosis. Okay, type 2 respiratory failure, like increase in carbon dioxide plus acidosis. Okay, and will cause a stimulation of the central, increase in carbon dioxide will cause a central chemoreceptors to stimulate and then cause the problem. Peripheral chemoreceptors, a decrease in O2 can cause the carotid body or the aortic bodies to get stimulated and cause the stimulation for the breathlessness feeling. Okay, so it's all like the feeling that you get from the respiratory center that and goes to the brain, and cortical awareness of a feeling of like a breathlessness. That means there is a problems over there. Okay, so respiratory diseases can stimulate breathing and dyspnea, stimulating intrapulmonary sensory nerves. Example: pneumothorax, interstitial inflammation, pulmonary embolus. Increasing the mechanical load can also cause this on the respiratory muscles. Example: airflow obstruction or pulmonary fibrosis. Okay, causing hypoxia or hypercapnia or acidosis. Stimulate the chemoreceptors. So all these like various parameters can end up in dyspnea. So one of the common complaint like breathlessness feeling. Okay. Okay, what are the some of the abnormal breathing patterns that we normally observe? Okay, so we have like two of them. One is the chain stocks, and the other one is the biots. Okay, Kussmaul breathing. There is another one that we have studied along with diabetes. Okay, diabetic ketoacidosis people will show the Kussmaul's breathing. Okay, so these two, like the chain stocks and the biots breathing patterns, are very serious conditions where like the end stage. Okay, when the patient is on an end stage condition, end stage respiratory problem, end stage cardiovascular problem, or an end stage nervous system problems. Okay, so in an end stage cardiovascular problem, you get chain stocks. Okay, chain stock, and in end stage central nervous system problem, you get Biot's breathing. Okay, so over here, because the cardiovascular system is not working, the tidal volume waxes and wanes. Okay, it increases, decreases, and then CA stoppage of breathing, again increase, decrease, and then stop. Okay, so this is the pattern that you see in the chain stock. Okay, accompanied by apnea. Okay, so the tidal volume increases, decreases, and then accompanied by apnea. So the blood O2 and the carbon dioxide fluctuate. So breath deeper, then smaller, then the tidal volume changes, respiratory rate can change too. Okay, so it will happen. So the reason for this is that because there is a problem in your cardiovascular system. What is happening? The oxygen carrying or the carbon dioxide coming to the central nervous system, the carbon dioxide of the respiratory neurons shows a difference on the carbon dioxide in the lung blood. Okay, so this gap, okay, is created. So because the blood is going slow from the heart to the brain. Okay, so the brain thinks, oh, there is a problem, and increases the respiratory rate. Okay, but when the brain increase the respiratory rate, you blow out a lot of carbon dioxide. After some time, that lack of carbon dioxide will reach to the brain. Okay, when it reaches to the brain, what will happen? The brain stops the ventilation because lack of carbon dioxide. We have seen before, when the partial pressure of carbon dioxide falls from 40 below to 35, what will happen? The ventilation will stop. That's why you get apnea. Okay, so there will be increase in the ventilation, then there is a decrease in the ventilation, and then there will be apnea, no ventilation. Okay, increase, decrease, and no apnea. So that is the pattern that is seen in the chain stroke. This is happening because the cardiovascular system, the pumping of the blood is slow from the lung, and that information of the change in the lung pattern to reach to the brain, it takes time because the heart is weak and pumping very slowly. Okay, another one, that is the Biot's breathing. The problem is in the CNS. Okay, so normal breathing pattern interrupted by periods of apnea. So you have increased breathing, then a period of apnea, again increased breathing, again a period of apnea. Okay, so this is switch on and off. Okay, on and off. So on off pattern that you see. Okay, so maybe a variant of chain stop breathing, so usually occur in patients with CNS disease. Okay, especially like meningitis condition, so where the respiratory center in the brainstem is affected. Okay, the respiratory center is affected over there, so the respiratory center is suddenly switching off and switching on, switching on and switching off. Okay, so that's why you see hyperapnea and then apnea, again increased breathing, and again stoppage of the breathing. Good. Okay, so that is about the two different types of abnormal breathing patterns. Okay, let's see another condition, that is the sleep apnea. Okay, so these are like two different types of sleep apnea. One is the central sleep apnea, and then the other one is the due to obesity and the peripheral sleep apnea. Okay, so sleep apnea due to classical reason is the obesity, the obese condition. Okay, due to lack of a very high amount of fat in the neck region, or due to weakness of the muscles in the neck regions. Okay, especially in the supine posture, the muscles weakness will cause or the rolling of the tongue or the compression of the air passages, nasopharynx, etc. will be blocked. Okay, so that's what happens in the case of sleep apnea. Okay, so what happens over here, you get snoring. Okay, so that is in the partial obstruction of airway. Okay, or complete obstruction of the airways can happen, and it can cause like serious effects. Okay, so in order to overcome that, we use the CPAP. Okay, so continuous positive airway pressure. Okay, so normal respiration happens like this. Okay, 0. Okay, so your drops inspiration and then expiration. So here you have like -1 and here you have like +1. Okay, so 0 is like 760. Okay, so at this neck region, if you keep like here inside also 760 and outside also 760, the chances of collapse is very high because the pressure, so because when it goes to -1, it is becoming 759. Okay, so when it becomes 759, what will happen, it will compress it. So for example, this is your neck. Okay, so this is your neck. Okay, so if you see here becomes 759 and it is 760 over here, what will happen, it will cause the collapse of it and the air coming over here will be blocked. Okay, so that's what happens in the sleep apnea. So in order to overcome this phenomena, what we use, the CPAP, the continuous positive airway pressure. So here it is 769 and here you never want to come below that. So how do we do that? We keep a pump over here. Here the airway pressure will be around like 765 always. Okay, so when you want to inspire, how, for example, this is your nose. Okay, this is like, okay, here it is now 765 and here above this is like 760. Okay, so for the inspiration to happen, the pressure will drop down from 765 to 764, but this is more than this 760. Okay, so no collapse will happen, the air will easily flow. In order for the air to go out, what should happen, that it 764 will change to 766 so that it can go out. So if I draw the graphical representation over here, instead of like this 0 like 760, 759, 761, you start operating at a higher length. Okay, so around 765. Okay, so less than that, more than that. So here 764, what will happen, inspiration, and 766, what will happen, you will have expiration. So this is happening only at the in front of the nose, like, so the respiratory system thinks the atmospheric pressure is 765, but the actual atmospheric pressure is only 760, so you don't get the compression. Okay, so this is like the modern CPAP machine. You just keep here and maintain the higher pressure over here so that you can inspire and expire very easily. Okay, so you operate at like around okay +5, not at 0 level. Okay, so that is the function of the continuous positive airway pressure. Okay, so that's how the CPAP machine works, so that you can like does not cause the obstruction. But this is not like useful for the central sleep apnea. Okay, central sleep apnea is due to not like an obstructive sleep apnea. It's a central sleep apnea is due to a problem within the brainstem and the respiratory regulatory centers. Okay, so like in the case of like a central hypoventilatory syndrome, etc. Okay, other machines that we use is the assisted mode ventilation, ACMV. This is like inspire, like see like like normally referred to as a ventilator. Okay, so inspiratory cycle initiated by the patient or automatically. Okay, that means if the patient is not starting, then the machine blow the air. Okay, if no signal is detected within a specified time window, expiration is then what will happen, the machine will cause the inspiration. Expiration is not assistant because that is passive, just stop that. Okay, positive and expiratory pressure, PEEP. Okay, that is used in like some condition like ARDS, AS, emphysema, etc., where you don't want the small alveoli to collapse. Okay, so that means the positive pressure is applied at the end of the expiratory cycle to decrease the alveolar collapse. Okay, used in condition like ARDS. Okay, or in like even for the emphysema, because like there are small alveoli that can collapse over there also, even though the lung is expanded over there. In the ARDS, there is no surfactant, so the chances of alveoli collapse will be more. Okay, so also you can ask the patient to do the pursed lip breathing. What happens in the pursed lip? You blow the air and bring it and keep it in the mouth, and then it is like positive pressure, and then you try to expire slowly through the nose. Okay, and during inspiration you can remove the air, the pressure is created, so that in the end of the expiration there is a pressure which will prevent the collapse of the alveoli. Okay, that's the PEEP. Okay, so let's discuss a little bit of like disease conditions we have been discussing before, okay, in a little bit more detail. Okay, so the common respiratory conditions, okay, for the diagnostic categories. Okay, so for the easy purpose, like to understand the pathophysiology, we have divided them into obstructive versus restrictive. Within restrictive there are two, parenchymal and extra parenchymal. Okay, obstructive conditions like asthma, chronic obstructive lung disease, chronic bronchitis, COPD, okay, which includes chronic bronchitis and emphysema coming together, or separately you can have chronic bronchitis and emphysema. For example, you can have emphysema alone in alpha 1 antitrypsin condition, alpha 1 antitrypsin deficiency. Okay, yeah, when you have alpha 1 antitrypsin deficiency, you can have emphysema alone without chronic bronchitis. Smokers normally get what? Chronic bronchitis with emphysema. That is why it is referred to as the chronic obstructive lung disorders, or referred to as the COPD. Okay, chronic obstructive pulmonary disease, okay, or bronchiectasis, okay, and cystic fibrosis, bronchiolitis. Okay, these are all like conditions in which you get difficulty for expiring. Okay, the air is not going out easily of the lung. Okay, so the air is getting trapped. Okay, so that is the meaning of the word obstructive lung disorder. Restrictive means what? Difficulty to inspire, the lung is unable to expand, so you have difficulty to expand, inspiratory problem. So obstructive lung disorder means it's an expiratory problem, and restrictive means what? Inspiratory problem. Inspiration is the problem over here. Okay, parenchymal means what? The lung itself, the lung tissue have the problem, like sarcoidosis, pulmonary fibrosis, pneumoconiosis, silicosis, asbestosis, etc. Okay, or rug or radiation induced interstitial lung disease. For example, one of the classical drug that can cause pulmonary fibrosis is amiodarone. Amiodarone used for the atrial fibrillation, etc. Okay, the cardiovascular conditions. Okay, amiodarone. We don't want to discuss more about the amiodarone that you will study in your pharmacology. Okay, so some drugs can induce these problems like pulmonary fibrosis as a side effect of it. Okay, that's the meaning. Okay, so other conditions that are extra parenchymal, not due to the problem of the lung. Okay, it's due to the muscles or the bones and the tissue, the outside the tissue problems, like neuromuscular. Okay, so diaphragmatic weakness or paralysis, damage to the cervical region, and damage to the phrenic nerve, etc., can cause diaphragmatic weakness or the paralysis of the diaphragm, or neuromuscular junction disease, which is like a myasthenia gravis, Lambert-Eaton, etc., Guillain-Barré syndrome, muscular dystrophies, cervical spine injury, etc. Okay, cause difficulty in the breathing. So that is all causing what? Inspiratory problem. You cannot inspire when the diaphragm is weak. Okay, and then the chest wall problem. The chest is unable to expand properly, like in the kyphosis or scoliosis, or the kyphoscoliosis, in obesity, ankylosing spondylitis of the neck. Okay, so all these regions, like when you have like stiffness or something, unable to expand. So this all causes what? Restrictive, inspiratory problem. Okay, extra parenchymal. So this is how we divide the diseases. Okay, especially in the pathophysiology, to understand how to distinguish between the conditions. Okay, one of the parameter that we use to distinguish between them is the peak expiratory flow, expiratory airflow. Here we check the speed of the air that is going out, the velocity by which the air is going out, how fast the air is going out. Okay, so in the normal, what happens, this on this you have the lung volumes. Okay, at this point is the total lung capacity, here is the residual volume. Okay, so residual volume. So from that, when you are expiring, okay, when you are expiring, from the total lung capacity to the residual volume, you are expiring. Okay, you're expiring. Okay, so you can see when you start expiring, suddenly the speed of the air, here is the speed of the air, liter per minute. Okay, the speed of the air. So suddenly what happens to the speed of the air? it rises, and then as the volume is declining, your speed of air is decreasing. Okay, so this is because when you are expiring, what happens to the airway passages? All of them is constricting. Okay, all of them is constricting. So because it is constricting, the air cannot easily flow out. So what will happen? The speed of the air that is flowing out will be decreased. So that is what you see. The speed of the air is declining. Okay, so here if you draw, it will be like this, and then it comes down. And for the inspiration, it goes like this. Okay, but this part of the loop is like, this is the inspiration and this is the expiration. Okay, so this part of the inspiration is not so important for the exam point of view. It is very useful when in the pulmonologist level they use this part also, but for the step 1 level you study this part, the expiratory loop. Okay, expiratory airflow. Okay, the expiratory is very useful for the diagnostic purpose. Okay, so now what happens in the case of obstructive, that this is the total lung capacity. Okay, the obstructive lung disorder, what happens to total lung capacity? That is increased. You get it from here. In the case of restrictive lung disorder, what happens to total lung capacity? It is decreased. You get it from here. Okay, and in the obstructive lung disorder, what happens to the residual volume? Is increased, whereas in the case of restrictive, residual volume is decreased. Okay, so this is how you get it in the case of restrictive. This is your restrictive condition. This will be the case of your obstructive. Okay, this will be the obstructive. Not only like this, in the case of obstructive, what you see, it goes high and then it comes down. It cannot go straight like this. There is no uniform decrease in the speed. Instead, it will show like this. Instead, it will fall initially and then drip and like goes like this. So this dip will happen in the case of obstructive. Okay, so because there is like the air passages are not easily allowing the air to go out, like in the case of asthma, because of the bronchoconstriction, as in the case of like chronic bronchitis, where there is a chronic bronchitis, mucus and other substance accumulating in the bronchus and the air is not able to go out. So as you start to expire, what happens to the speed? Suddenly drops and then it goes slowly out. It takes a longer time for the air to go out and it slowly goes. So this dip that you can see is a very characteristic of the obstructive. See over here, you can see this dip. Okay, so this is your airway obstructive lung disorders, and this is restrictive lung disorders. Constricted lung means restrictive lung disorders. Okay, so this is how you can distinguish between the obstructive and restrictive lung disorder by looking at the peak expiratory airflow. Okay, another one, like small like information about how to distinguish between parenchymal and extra parenchymal restrictive diseases. Okay, so one of the classical thing that you can see over here in the parenchymal problem, like for example like fibrosis, etc., what happens to the residual volume? Residual volume is decreased. Okay, the residual volume is decreased over here. The residual volume is decreased, the RV, the residual volume is decreased in the case of fibrosis. But in extra parenchymal, you won't see a decrease in the uh, residual volume. Okay, so that is one of the criteria you can use. But classically, you just have to exam point of view mainly concerns about this fibrosis and obstructive, and this dip that you see over here. This dip that you see over here. These are the important points that you can keep in your mind. Okay. Okay. So by this information, let's look into this question over here. Okay. Okay. So a 65-year-old female is seen in clinic for chronic cough. She has a productive cough for most of the past 2 years. She also complains of newly worsening exertional dyspnea over the last 3 months. She has no medical history and takes no medication, though she has not assessed healthcare in 5 years. She has smoked a pack of cigarettes a day for the past 50 years. Okay, and state that she is unwilling to quit. Okay, so cigarette smoking is the one of the important clue that is given to you. Okay, and vital shows temperature, blood pressure, okay, pulse, respiratory rate, oxygen saturation is okay, and she is elderly woman who is quite thin. Okay, her ribs are visible and there is hypertrophy of the intercostal muscles. She emits scattered expiratory wheezes and has decreased aeration diffusely. She breathes through her pursed lips with a prolonged expiration phase. Okay, you can see the prolonged expiratory phase. So it takes a long time for the expiration to happen. Okay, that means it is the air is not going out fast, so expiration is difficult for her. Okay, and her heart is regular and there is no peripheral edema. A chest X-ray shows a hyperinflated with the diaphragm flattening and a small heart. Okay, because the lung is hyperinflated, so the heart will be like restricted over there. PaO2 and the arterial blood gas trust is 64. PaCO2 is 60, but a normal pH. Okay, so you can see oxygen is dropped and carbon dioxide is higher. Okay, she is suspected of COPD, so the diagnosis is given to you. Okay, and is sent for the pulmonary function testing. Okay, so the diagnosis is given to you. So now, the which of the following closely represents the patient's flow volume loop? Okay, so this is the question. Okay, expiratory flow rate. Okay, so as we have discussed, there should be this dip over here. So this does not show the dip, this is not showing the dip, this is showing the dip. These two are showing the dip. Okay, so either say option A, this is B, this is your C, this is D, this is E. So you crossed out C, D and E. Okay, now you are left with A and B. Okay, so now in the obstructive lung disorder, what should happen to the total lung capacity? Okay, the total lung capacity is more and residual volume is more. Okay, increased. Okay, so in out of these two, which one shows the increased residual volume? Okay, over here you can see the residual volume is high. Okay, the total lung capacity is high. Okay, residual volume is high, whereas here the residual volume is very less and the total lung capacity is very less. Okay, so this is not the option. Okay, so this is your correct option. Okay, so this is what you have to pick for in this condition. Okay, represents this patient's flow volume loop. Okay, okay, at least in this question it's good that your diagnosis is given to you. Even without that, a prolonged expiratory problem, okay, and a flattened diaphragm. Okay, so this flattened diaphragm that is over here, the flattened diaphragm is a very good clue. Okay, and also another clue is cigarette smoking for like the past 50 years. Cigarette smokers have a high risk to get COPD. Good. Okay, now in order to distinguish within the obstructive, okay, now it is pretty easy to distinguish between obstructive versus restrictive. Within the obstructive, to distinguish between asthma versus COPD. Okay, the asthma versus COPD. Okay, we need to know about the diffusion capacity. Okay, in asthma the diffusion capacity of the lung is not affected. Okay, which is normally referred to as either TLCO or KCO. So we use the carbon monoxide to measure the diffusion capacity of the lung, DLCO. This is one of the classical thing that is used to distinguish between them. Okay, so you can see in the asthma what happens to the diffusion capacity. In the asthma, no change. Okay, in the chronic bronchitis also no change, but in emphysema it is markedly decreased. In fibrosis it's markedly decreased. Okay, why in chronic bronchitis? It's a bronchus problem, it's not a problem with the alveoli. Okay, so you know the diffusion equation. Diffusion is directly proportional to delta P, area divided by the thickness, and the solubility divided by root of molecular weight. This is a constant. Okay, we keep this as a constant over here. Okay, so what is the, changing over here? The area. In emphysema, what happens to the area? The area is decreased. Okay, so when you have area is decreased, what happens to diffusion? Diffusion decreased. Okay, in pulmonary fibrosis, what happens to thickness of the membranes? The thickness that is represented here by the D, the thickness increases. So when there is an increase in thickness, what happens? The diffusion decreases. Okay, so that's why you will see marked decrease in the pulmonary fibrosis and in emphysema. Whereas if you have only chronic bronchitis, no problem in the diffusion. If you have asthma, no problem in the diffusion. Okay, that's what you will see. Okay, in all these conditions FEV1 will be decreased. All these conditions the FEV1 is decreased. FVC, the vital capacity, is decreased in all the conditions. FEV1 by FVC ratio is decreased in chronic diseases but increased or normal in this pulmonary fibrosis. This is your restrictive condition. Okay, so that's one of the classical thing that is used to distinguish between obstructive versus restrictive, FEV1 by FVC ratio. Okay, and then what happens to the total lung capacity? Total lung capacity increases in obstructive, whereas in the restrictive it is decreased. Okay, another important thing, residual volume is increased in all these, okay, but decreased, it's increased over here in the obstructive, whereas decreased in the case of restrictive lung disorder like fibrosis. In asthma, the one important thing, there may not be a sufficient increase in total lung capacity or in the residual volume. Okay, depends on the duration of the asthma. Okay, so it may not be seen, that phenomena may not be seen. Okay, whereas in the chronic bronchitis emphysema, it's very evident. The total, especially in emphysema, you can see the barrel chest and the lung capacity will be increased. Okay, so very important in the exam point of view, you have to remember about what happens to your diffusion capacity, which is represented by DLCO, carbon monoxide diffusion capacity, because carbon monoxide is the most difficult gas to diffuse across. Okay, it has the most least solubility compared to oxygen, carbon dioxide. It has the least. Oxygen is having 20 times less solubility than carbon dioxide. Carbon dioxide has the highest solubility, and the carbon monoxide have less solubility than the oxygen also. So that's why we use the carbon monoxide to understand the diffusion capacity. Okay, so the some other this the way of representing the graphs. Okay, this one that you see over here is the block in your trachea, or like some the airways are blocked, so you get uniform speed of the airway during inspiration and during expiration. This dip over here will tell you this is the obstructive, and this is the restrictive. Okay, the restrictive decreased. Okay, here also, here you take the FEV1 and FVC on this axis. Okay, FVC, and then you have the time. Okay, you have the time over here. So if this is your normal, in both restrictive and in the obstructive, what is happening, it is decreased. FVC is decreased, and FEV1 is like very much increased. In 1 second you cannot have, like, it's decreased, but the duration, the time taken for the whole expiration to happen, okay, is very much increased. So this is your obstructive. It's very short compared to the normal. Okay, this is the normal time, the time taken is over here. But for the restrictive, so this is your restrictive condition. Okay, the different ways of representing this also we have seen. Okay, like the spirometry. So this is high over here, this is your obstructive, this is down over here, this is your restrictive. Okay, and the time taken for here is a long time taken for the expiration to happen. Okay, so this is your obstructive problem, and this is restrictive. So various ways by which representing this graphical representation, very important for your exam point of view. It keeps coming up in your exams, the graphs. Okay, the pulmonary function test. Okay, some specific pulmonary abnormality. Okay, like for example, how in comparison the pneumonia versus atelectasis or pneumothorax. Okay, now what happens to the partial pressure of oxygen in the blood? Okay, so here you can see pneumonia is an inflammatory condition of the lung in which the alveoli are filled with fluid and blood cells. Gas exchange functions has progressively declined because of the thickness increasing and fluid accumulation. The exchange of the gas is very much decreased. So reduction in the available surface area of the respiratory membrane. Okay, so decreased ventilation perfusion ratio will be seen. Okay, decreased ventilation perfusion ratio, because perfusion is coming there, but ventilation is there, but it is unable to cause the exchange. Okay, ventilation is not able to exchange. Okay, in the atelectasis, collapse of the alveoli, localized air of the entire lung. You will see airway obstruction, blockage of many bronchi with mucus, or obstruction in the major bronchus with mucus or tumor. Okay, and lack of surfactant that's causing the lung collapse in premature babies, and surface tension of the alveoli fluid increases dramatically. Okay, now let us see over here. So the blood is coming from here. Okay, the blood that comes. Okay, how much percentage is like blood goes here, like no, the blood is getting saturated 97 percentage, because in this lung almost like 50 percentage, half of the blood goes here and half of the blood is going here. Okay, but here only 60 percentage is saturated because of the pneumonia. The air come in here, but the air is unable to exchange. Okay, so you get only 60 percentage saturation, but the air that has gone to this is the normal lung. Okay, this is the normal lung, this is the normal lung, and this is the lung with pneumonia. In the normal lung you get the normal saturation. Okay, so on an average, what happens? Half of the blood is normal saturated, half of the blood is only 60% saturated with oxygen. So the mean oxygen saturation is decreased over here. Okay, whereas over here you can see this is the lung with atelectasis and this is the normal lung. Okay, the normal lung, this is the collapsed lung. Okay, what happens to the blood supply? The blood that goes over here, the major, almost like 90% of the blood goes here, and only the 10% of the blood is going over here. Okay, so even though the blood the air that comes over here is less and there is 60% only can be exchanged, but here you can get like, but the good lung have, the normal lung have what, the majority of the blood going. So the majority of the blood, or almost 5/6 of the blood, is getting good saturation. Only like the 1/6 of the blood is like partially saturated. So on overall, the saturation, the oxygen saturation of the hemoglobin, oxygen saturation in the blood, the oxygen content in the blood will be good. Okay, this is almost equal to the normal percentage. Okay, normal 97% saturation, you can see. Okay, so that is the one classical difference between pneumonia and atelectasis. Okay, in pneumonia there will be a major decline in the partial pressure of oxygen. Okay, or the oxygen saturation in the blood, or the oxygen content in the blood will be markedly dropped compared to that of a pneumothorax or atelectasis. Okay, now let's discuss this little bit about hypoxia. Okay, hypoxia. Okay, one important point I want to mention about the cyanosis. Cyanosis is caused by the deoxygenated hemoglobin. Deoxygenated hemoglobin. Okay, the deoxygenated hemoglobin gives the blue color. The blue color. But if there is no hemoglobin, like in the case of anemia, you can't get the blue color. Okay, if you don't have hemoglobin, you don't get the blue color. Okay, it is pale. Okay, you cannot show the red color, the redness, as well as no blue color, because oxygenated blood is red in color, deoxygenated blood is blue in color. If you don't have the hemoglobin, like in anemia, you cannot show the cyanosis. So what you observe when you look at the eyes or somewhere, it will be pale in color, not blue in color. Okay, so cyanosis. Okay, so that is why in anemic hypoxia caused due to anemia, what will not be seen, you will not see cyanosis. Okay, in histotoxic hypoxia, that means the tissues are unable to use the oxygen, you will not see the cyanosis, because tissues are not able to use the oxygen. That means oxygen stays in the blood, you will not get the deoxygenated hemoglobin. Okay, it will be always oxygenated. Okay, so in anemic hypoxia, in stagnant hypoxia, that means the cardiovascular, the blood is unable to pump, or there's a block somewhere and the blood is unable to go. Okay, so what happens to the arterial partial pressure of oxygen? Arterial partial pressure of oxygen, no change. Okay, it stays the same. Okay, anemic hypoxia is the dissolved oxygen, very important, keeps coming in the exams. Okay, anemic hypoxia, what happens to the arterial PO2? No change. Why there is no change? Because the dissolved oxygen is the one that determines the PO2. The partial pressure is determined by dissolved oxygen. And anemic hypoxia, there is no problem for the dissolved oxygen. Okay, okay, that's one. Okay, now let's look at the arterial PCO2, hypercapnia. Okay, not so relevant over here. It does not increase, does not show much change. Okay, okay. In the case of like stagnant hypoxia, sometimes you can get cyanosis. Okay, you can get cyanosis because the blood is stagnated. Say for example, stagnated in the arm, all the blood will take the oxygen from the hemoglobin, so hemoglobin will turn the color into blue because it becomes deoxygenated. So the blood that is stagnated in one region will show bluish discoloration. That's why cyanosis. The oxygen therapy is of no use. Okay, in all these conditions, the oxygen therapy, even if you give oxygen, for example, anemic patient, there is no hemoglobin to carry the oxygen, so even if you give the oxygen, there is no much oxygen that can be carried by the blood. That's why the oxygen therapy is of not much useful. Okay, so all this, the remaining hypoxia is called as the hypoxemic hypoxia, or the arterial hypoxia. That means lack of oxygen in the blood. Hypoxemia means lack of oxygen in the blood. Hypoxia means lack of oxygen to the tissues. So hypoxemic hypoxia. So lack of oxygen to the tissues due to lack of oxygen in the blood. Okay, that's the meaning. So how can you get lack of oxygen in the blood? Either due to hypoventilation, the person is not breathing, or the person has gone to high altitude, because the partial pressure of oxygen is very less in the case of high altitude. So or diffusion limited, like fibrosis, etc., where the diffusion is not happening. Okay, the gas is coming but unable to diffuse to the blood. Okay, or the physiological shunt. Okay, V by Q mismatch, anatomical shunt, congenital, or the cardiac problems. Okay, the blood is not able to receive the oxygen. Okay, so what happens to the arterial PO2, arterial partial pressure of oxygen will be dropped. Okay, arterial partial pressure of oxygen will be dropped because the blood is not able to get the oxygen. So the dissolved oxygen will be dropped in the blood. Okay, some carbon dioxide depends on the duration of the disease condition. Okay, most of the condition, the duration, because carbon dioxide is something like have a 20 times more solubility than oxygen. Okay, so it is easy to exchange, or it won't accumulate too much. For example, in fibrosis, unless the fibrosis is very severe, what will happen, you won't have a too much of accumulation of carbon dioxide. So especially the hypercapnia is depending on the duration of the disease condition. And cyanosis, all these conditions, you have sufficient blood, you expect to have sufficient blood and hemoglobin to be like normal. Okay, and then in that scenario, what will happen, when there is lack of oxygen, the hemoglobin does not get sufficient oxygen, so the hemoglobin will become deoxyhemoglobin and will show cyanosis. Oxygen therapy is of useful in this condition, but has to be used with caution, like because oxygen is highly toxic to the body. Okay, so if the oxygen is less, so if you want, like for example in fibrosis, even if you give oxygen, because just now we have seen the diffusion depends on the delta P, you can increase your delta P and cause the diffusion to happen. Okay, so sometimes it will be useful depending on the situation. Okay, so that's about the important points about the hypoxia. Okay, so with this we conclude the respiratory physiology. Okay, in the next topic we continue with the acid-base balance, in which the respiratory system also take an important role. Okay, so for this, for now, we conclude the respiratory physiology.